Within the heart of every living cell lies a microscopic marvel, a molecule so profound that it dictates the very essence of life itself, deoxyribonucleic acid, or DNA. This molecule, though incredibly tiny, holds the key to understanding the biological blueprint of all living organisms. It is the fundamental building block that carries the genetic instructions used in the growth, development, functioning, and reproduction of all known living organisms and many viruses. DNA is the thread that weaves the fabric of life, connecting every living being in a complex web of genetic information. This intricate molecule serves as the blueprint of life, carrying within its spiraling strands the genetic instructions that shape every organism on Earth, from the smallest bacterium to the towering oak tree. The double helix structure of DNA, with its two strands coiled around each other, is a marvel of nature's engineering. Each strand is composed of a long chain of nucleotides, which are the building blocks of DNA. These nucleotides pair up in specific ways, forming the rungs of the twisted ladder that is the DNA molecule. The sequence of these nucleotides encodes the genetic information that determines the characteristics of an organism. DNA holds the secrets to our inheritance, passed down through generations, connecting us to a vast and ancient lineage of life. It is through the transmission of DNA from parents to offspring that traits are inherited and the continuity of life is maintained. This genetic inheritance is what makes each species unique, yet it also binds all living organisms together in a shared history. The study of DNA has allowed scientists to trace the evolutionary history of species, uncovering the connections between different forms of life and revealing the story of life's diversity on Earth. Imagine DNA as an elaborate instruction manual, a detailed recipe book for life. Each page of this book contains specific instructions for building and maintaining an organism. These instructions are written in the language of nucleotides, with each sequence of nucleotides representing a different piece of information. Just as a recipe book contains instructions for making different dishes, the DNA instruction manual contains the information needed to create the various proteins and molecules that make up an organism. This genetic code is universal, shared by all living organisms, and it is the basis for the incredible diversity of life on Earth. This manual contains all the information needed to build, maintain, and replicate an organism. The process of DNA replication is a critical part of cell division, ensuring that each new cell receives an exact copy of the genetic information. This precise duplication of DNA is essential for growth, development, and repair in living organisms. Scientists have made remarkable strides in understanding the mechanisms of DNA replication, and this knowledge has paved the way for advances in genetic engineering, biotechnology, and medicine. By manipulating the genetic code, researchers can develop new treatments for diseases, improve crop yields, and even create new forms of life. It determines our physical characteristics, influences our susceptibility to diseases, and even plays a role in shaping our personalities. The genetic code within our DNA is responsible for the traits that make us who we are, from the color of our eyes to our height and even our predisposition to certain health conditions. Advances in genetic research have led to a deeper understanding of how genes influence our health and behavior. By studying the genetic variations that contribute to diseases, scientists are developing personalized medicine approaches that tailor treatments to an individual's genetic makeup, offering the promise of more effective and targeted therapies. Understanding DNA is akin to unlocking the secrets of life itself, revealing the intricate mechanisms that govern our existence. The study of DNA has revolutionized biology, providing insights into the fundamental processes of life. By decoding the genetic information within DNA, scientists can uncover the molecular basis of diseases, understand the genetic factors that contribute to health and development, and explore the evolutionary relationships between species. This knowledge has profound implications for medicine, agriculture, and conservation, offering new ways to address some of the most pressing challenges facing humanity. The discovery of DNA's structure in 1953 by James Watson and Francis Crick, with crucial contributions from Rosalind Franklin, revolutionized the field of biology. Their groundbreaking work revealed the elegant double helix structure of DNA, a twisted ladder-like molecule composed of two intertwined strands. This discovery was a monumental achievement, providing the foundation for modern genetics and molecular biology. Rosalind Franklin's X-ray diffraction images were critical in elucidating the structure of DNA, and her contributions have since been recognized as pivotal to this scientific breakthrough. Their groundbreaking work revealed the elegant double helix structure of DNA, a twisted ladder-like molecule composed of two intertwined strands. This structure explained how DNA could replicate and carry genetic information, providing a molecular basis for inheritance. The double helix model has become an iconic symbol of biology, representing the unity and diversity of life. The discovery of the DNA structure has had far-reaching implications, leading to the development of new technologies and techniques for studying and manipulating genetic material. This discovery opened up a whole new world of scientific inquiry, paving the way for advancements in medicine, agriculture, and our understanding of evolution. The ability to sequence and analyze DNA has transformed our understanding of the genetic basis of diseases, leading to the development of new diagnostic tools and therapies. In agriculture, genetic engineering has enabled the creation of crops with improved yields, resistance to pests, and enhanced nutritional content. The study of DNA has also provided insights into the evolutionary history of life on Earth, revealing the genetic connections between species and the processes that drive evolution. As we continue to explore the mysteries of DNA, we unlock new possibilities for improving human health, enhancing food security, and understanding the natural world. DNA resides within the nucleus of every eukaryotic cell, acting as the central repository of genetic information. This nucleus is like the command center, safeguarding the instructions that dictate the cell's functions and characteristics. Neatly packaged into structures called chromosomes, DNA is organized in a way that ensures its stability and accessibility. These chromosomes are essential for the proper distribution of genetic material during cell division. Think of chromosomes as carefully organized volumes within the vast library of DNA. Each volume contains a wealth of information, meticulously arranged to ensure that the cell can access the right instructions at the right time. Humans possess 23 pairs of chromosomes, each containing thousands of genes. These genes are the blueprints for our physical and functional traits, from the color of our eyes to the way our bodies metabolize food. Genes are specific segments of DNA that code for particular traits, dictating everything from eye color to height. They are the fundamental units of heredity, passed down from generation to generation. Prokaryotic cells, such as bacteria, lack a true nucleus but still rely on DNA for their genetic information. In these simpler organisms, the DNA is not enclosed within a membrane-bound nucleus. Their DNA exists as a single circular chromosome, free-floating within the cell's cytoplasm. This arrangement allows for the rapid replication and transfer of genetic material, which is crucial for their survival and adaptation. Despite the differences in organization, the fundamental role of DNA remains the same across all life forms: to store and transmit genetic information. This universal function underscores the shared ancestry of all living organisms. DNA is not a static entity. It is actively involved in various cellular processes. It plays a dynamic role in the synthesis of proteins, which are essential for the structure and function of cells. One of its primary functions is replication, the process of creating an exact copy of itself. This ensures that genetic information is accurately passed on during cell division, maintaining the continuity of life. This duplication is essential for cell division, ensuring that each new cell receives a complete set of genetic instructions. Without this precise replication, cells would not be able to function properly or sustain life. Without DNA replication, life as we know it would cease to exist. This fundamental process is the cornerstone of biological continuity, enabling growth, development, and evolution across generations. To truly grasp the complexity of DNA, we must delve into its fundamental building blocks, nucleotides. These tiny molecules are the essential units that make up the DNA strand, and understanding them is key to unlocking the secrets of genetic information. Imagine nucleotides as the individual letters in the genetic alphabet. Just as letters combine to form words and sentences, nucleotides come together to create the instructions for life. Each nucleotide consists of 3 components: a sugar molecule called deoxyribose, a phosphate group, and a nitrogenous base. The deoxyribose sugar is a 5-carbon sugar that forms the central framework of the nucleotide, while the phosphate group links the sugars together, creating a sturdy backbone. The sugar and phosphate groups form the backbone of the DNA strand, providing structural support. This backbone is like the spine of a book, holding everything together and giving the DNA its characteristic double helix shape. The nitrogenous bases, on the other hand, are the true carriers of genetic information. These bases pair up in specific ways to encode the instructions needed for building and maintaining an organism. There are 4 different types of nitrogenous bases in DNA: adenine, A, thymine, T, guanine, G, and cytosine, C. Each of these bases has a unique structure and function, and they pair up in a very specific manner. These 4 bases, arranged in specific sequences along the DNA strand, form the genetic code. This code is like a set of instructions that tells the cell how to build proteins, which are the building blocks of life. The sequence of these bases determines the specific instructions encoded within a DNA molecule. Just as the order of letters in a sentence determines its meaning, the order of bases in a DNA strand determines the genetic information it carries. For instance, a sequence of ATCG might code for a particular protein involved in eye pigmentation, while a sequence of GCTA could code for a protein involved in muscle function. These sequences are read by the cell's machinery and translated into the proteins that perform various functions within the organism. The vast number of possible base combinations allows for the incredible diversity of life we see on Earth. From the simplest bacteria to the most complex plants and animals, the unique sequences of DNA in each organism encode the instructions that make life possible. This diversity is a testament to the power and versatility of the genetic code. Now that we understand the building blocks, let's envision how these nucleotides come together to form the iconic double helix structure of DNA. Picture a ladder twisted upon itself, forming a spiral staircase. The sugar-phosphate backbones of the 2 DNA strands form the sides of the ladder, while the nitrogenous bases protrude inwards, forming the rungs. The 2 strands of DNA are not identical but rather complementary to each other. This means that the sequence of bases on one strand dictates the sequence of bases on the other strand. The bases pair up in a very specific manner. Adenine, A, always pairs with thymine, T, and guanine, G, always pairs with cytosine, C. This complementary base pairing is crucial for DNA replication and the transmission of genetic information. When DNA replicates, the 2 strands separate and each strand serves as a template for the synthesis of a new complementary strand. This ensures that each new DNA molecule is an exact copy of the original, preserving the genetic code accurately.